Plant kingdom. The earlier systems of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They were mainly based on vegetative characters or on androecium structure, system given by Linnaeus. Such systems were artificial. They separated closely related species since they were based on a few characteristics. Also, the artificial systems gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification systems developed, which were based on natural affinities among the organisms and considered not only external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time, hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy that uses the chemical constituents of plant to resolve confusions are also used by taxonomists these days. Algae, further divided into chlorophyce, pheophyce, and rhodophyce. Algae are chlorophyll bearing, simple, thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with fungi as lichens and animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous forms like Eulothrix and Spirogera. A few of the marine forms, such as kelps, form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by production of different types of spores, the most common being the zoospores. They are flagellated, motile, and on germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, or non-flagellated and non-motile, but similar in size as in Spirogera. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed as oogamous, example Volvox and Fucus. Figures showing green algae, Volvox and Eulothrix. Volvox has daughter colony, parent colony. Eulothrix, cup-shaped chloroplast, hold fast, pyrenoids, and Spirogyra. Eulothrix is found in fresh water, running fresh water, and Spirogyra in stagnant fresh water. Brown algae, Laminaria. Frond, stipe, hold fast. Frond is leaf-like photosynthetic. Stipe is stalk and hold fast. Fucus has air bladder, frond, midrib, stipe, and hold fast. Dictyota has frond and two branches occur from each branch. So Dictyota. Laminaria, Fucus, Dictyota are brown algae. Red algae, Porphyra and Polysiphonia. Porphyra has frond, Polysiphonia has main axis, branches. Algae are useful to man in a variety of ways. At least a half of total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of food cycles of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrocolloids, that is, water holding substances. Example algin by brown algae and carrageen red algae, which are used commercially. Algin brown algae, carrageen red algae. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, unicellular alga rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplast. Chloroplast may be discoid plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and outer layer of pectocellulosic cell wall. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eleutheryx, Pyrogera, and Cara. Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms, ectocarpus, to profusely branched forms, as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment, fucoxanthin, present in them. Food is stored as complex carbohydrates, which may be in form of laminarin or mannitol. Vegetative cells have a cellulosic wall, usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to a substratum by a holdfast and has a stalk, the stipe, and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two equal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogonium in oogamous species. The gametes are pyriform, that is pear-shaped, and bear two laterally attached flagella. Common forms are Ectocarpus, Dictata, Laminaria, Sargassum, and Fucus. Members of Rhodophyceae are commonly called red algae because of the predominance of red pigment R-phycoerythrin in their body. Majority of red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as fluoridine starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysiphonia, Porphyra, Gracilaria, and Gelidum. Divisions of algae and their main characteristics. Chlorophyceae, common name green algae. Major pigments are chlorophyll A and B. Food stored as food stored in the form of starch, pyrenoid in chloroplast, and also oil droplets in protoplast. Cell wall is of pectocellulosic, pectin, and cellulose. Flagellar number and position of insertions 2 to 8 equal apical flagella. 2 to 8 equal apical flagella. Present in fresh water, brackish water, and salt water. Phaeophyceae brown algae, chlorophyll A, C, and fucoxanthin. Food stored as mannitol and laminarin. Cell wall is made of cellulose and algin. Two flagella, unequal and lateral, piriform gametes. Freshwater, rarely found in freshwater, brackish water, salt water. Rhodophysi, red algae, chlorophyll A, D, phycoerythrin. Food stored as fluoridine starch similar to amylopectin and glycogen in structure. Cell wall is cellulose, pectin and polysulfate esters. Flagella absent. Some are present in fresh water, brackish water, mostly in salt water. In fungus, food is stored in the form of glycogen. Bryophytes, amphibians of plant kingdom, includes liverworts and mosses. Figure 3.2, bryophytes, a liverwort marchantia, female thallus and male thallus are shown. Gametophytes. Okay, so female thallus include archegonium, which is flask shaped and produce a single egg, archegoniophore, gamma cup, which bear gamete. Rhizoids may be unicellular or multicellular. It's a liverwort. Male thallus include anthridium, which produce biflagellate anthrazoids, anthridiophore, gamecup, rhizoids. Mosses, Panaria, gametophyte and sporophyte are shown. Sporophyte is 2N, includes capsule, which contains spores after meiosis, seta and foot. The ploidy level is 2N for sporophyte. Ploidy for gametophyte is N, includes leaves, main axis and rhizoids. Rhizoids are multicellular and branched. Figure D, sphagnum. Gametophyte is shown. Anthridial branches, archegonial branch at bottom. Bryophytes include various mosses and liverworts, commonly growing in moist, shaded areas in the hills. Bryophytes are also called amphibians of plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in plant succession on bare rocks and soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to other substratum by unicellular or multicellular rhizoids. They lack true roots, stem or leaves. They may possess root-like, leaf-like or stem-like structures. The main plant body of bryophyte is haploid. It produces gametes, hence is called gametophyte. Sex organs in bryophytes are multicellular. The male sex organ is called anthridium. They produce biflagellate anthrazoids. Female sex organ called archegonium is flask-shaped and produces a single egg. Anthrazoids are released into water where they come in contact with archegonium. An anthrazoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a cellular body called a sporophyte. Sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packaging material for transshipment of living material because of their capacity to hold water. Mosses, along with lichens, are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats in the soil, they reduce the impact of falling rain and prevent soil erosion. Bryophytes are divided into liverworts and mosses. The main plant body of bryophytes is the gametophyte. N gives rise to gametes, which are also N. Gametes on fusion produce zygote, which is 2N. Zygote without meiosis give rise to sporophyte, which is 2N, a multicellular body. Sporophyte on meiosis produce spores, which are N, and spores on germination produces gametophyte. Sporophyte is dependent on gametophyte for food. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid, example Marchantia. Thallus is dorsiventral and closely appraised to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by formation of specialized structures called gamete, single gamma. Gamete are green multicellular asexual birds which develop in small receptacles called gamecops located on the thalli. Gamete become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Sporophyte is formed from zygote germination and spores after meiosis of sporophyte. Meiosis. The predominant stage of the life cycle of a moss is the gametophyte, which consists of two stages. First stage is the protonema stage, which develops directly from a spore. It is creeping, green, branched and frequently filamentous stage. Second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright slender axes bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs at apex. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of leafy shoots. After fertilization, zygote develops into a sporophyte consisting of a foot, seta, and capsule. Sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Pteridophytes include classes Psilopsida, Lycopsida, Phenopsida, and Pteropsida. First terrestrial because of xylem and phloem. The pteridophytes include hostels and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues like xylem and phloem. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. In bryophytes, the dominant phase is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissues. The leaves in pteridophyta are small, microphyll as in Selaginella, or large, that is macrophyll as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones, as in Selaginella and Equisetum. The sporangia produce spores by meiosis in spore mother cells. Spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of anthrozoids, the male gametes released from the antheridia, to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte, which is the dominant phase of pteridophytes. In majority of pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro, large, and micro, small spores, are known as heterosporous. Heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Xylotum, Lycopsida, Selaginella, Lycopodium, which has medicinal value. Lycopodium spores are edible. Phenopsida equisetum and Pteropsida Drypteris edientum. Gymnosperms. The gymnosperms, gymnos means naked spermacids. Gymnosperms are plants in which ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. Seeds that develop post fertilization are not covered, that is, are naked. Gymnosperms include medium sized trees or tall trees and shrubs. One of the gymnosperm, the giant redwood sequoia, is one of the largest tree plant species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, that is pinus, example pinus, while in some others, cycas, small specialized roots called coralloid roots are associated with entofixing cyanobacteria. The stems are unbranched as in cycas or branched as in pinus and cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. Gymnosperms are heterosporous. They produce haploid microspores and megaspores. Two kinds of spores are produced within sporangia that are borne on sporophylls which are arranged spirally along an axis to form lacs or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains take place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be born on the same tree as in Pinus. However, in Cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are born on megasporophylls which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms the male and female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on the megasporophylls. The pollen tube carrying the male gametes grows through the archegonia in the ovules and discharges their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. These seeds are not covered. Anemophily. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of ovules borne on the megasporophylls. That is wind pollination. Figure 3.4 Gymnosperms. First is cycas. Cycas, unbranched stem, pinnate leaves, persist for few years. Male and female cones born on different tree. Entofixing cyanobacterial association. Coralloid roots, that is entofixing cyanobacterial association. Figure B is pinus, branch stem, male and female cones born on same tree. Fungal association with mycorrhiza. Third figure is ginkgo. It's a living fossil. Labelings are dwarf shoot, long shoot, seeds. Figure 3.3 Pteridophytes. First figure is Selaginella. Microphylls, heterosporous. Labelings are stem, leaves, roots, and rhizophores, a specialized structure, organ sui generis, strobili or cone, equisetum, rhizome, it's also called horsetail. Labelings are strobilus, a compact structure, node, internode, branch, it's homosporous. Fern, homosporous, macrophyls, Drypteris teres are the examples. Salvinia, heterosporous. Selaginella and Salvinia are heterosporous. Strobili or cones formed in Selaginella and equisetum. Angiosperms. Unlike gymnosperms where the ovules are naked, in angiosperms or flowering plants, pollen grains and ovules are developed specialized structures called flowers. In angiosperms, seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from smallest wolfia to tall trees of eucalyptus over 100 meters. Smallest plant is Wolfia rhiza, tallest tree is Eucalyptus regnans. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, dicotyledon and monocotyledons. The dicots are characterized by seeds having two cotyledons, reticulate venation in leaves and tetramerous or pentamerous flowers, etc., that is, having 4 or 5 members in each floral holes. The monocots, on the other hand, are characterized by single cotyledonous seeds, parallel venation in leaves, and trimerous flowers having 3 members in each floral hole. The male sex organ in a flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anthers, the pollen mother cell divides by meiosis to produce microspores, which mature into pollen grains. The female sex organ in a flower is the pistil. Pistil consists of an ovary at its base, a long slender style and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and one divide to form the embryo sac. Each embryo sac has a 3-celled egg apparatus, 1 egg and 2 synergids, 3 antipodal cells, and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anthers are carried by wind on various other agencies to stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and resulting pollen tubes grow through the tissues of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with the egg cell syngamete to form a zygote. The other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus (PEN). Because of the occurrence of 2 fusions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. Zygote develops into an embryo with 1 or 2 cotyledons, and the pen develops into endosperm, which provides nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events, the ovules develop into seeds and the ovaries develop into fruit. Zygote develops into embryo, pen into endosperm, ovule into seed, ovary into fruit, and integument into seed coat. Life cycle of an angiosperm. The main plant body is sporophytic. So, male sporophyte, anther with filament. Anther consists of microsporangium, which on differentiation gives rise to microspore or pollen mother cell. Microspore mother cell or pollen mother cell. Microspore mother cell on undergoes meiosis to form 4 microspores. Microspores mature to form pollen grain. Pollen grain or microspore is the gametophytic generation, ploidy level N. Microspore mother cell or pollen mother cell, ploidy is 2N. Female Sporophyte is ovary, which has stigma and style. Ovary consists of ovule or megasporangium. Each ovule or megasporangium has a megaspore mother cell which undergoes meiosis to form 4 haploid megaspores, out of which 3 degenerate and 1 divide to form embryo sac. Each embryo sac has a 3-celled egg apparatus which consists of 1 egg cell and 2 synergids, 3 antipodal cells and 2 polar nuclei. Embryo sac is haploid or gametophytic generation. Megaspores ploidy level N. Megaspore mother cell is 2N. Now the male gametophyte, that is the pollen grain, after pollination germinate on stigma, form pollen tube, grows through the tissues of stigma and style and reach the ovule. Pollen tube enters embryo sac guided by finger-like projections of synergids and release its 2 gametes, male gametes, out of which 1 male gamete fuses with the egg cell called syngamy to form a zygote, and the other male gamete fuses with diploid secondary nucleus to produce a triploid primary endosperm nucleus PEN, which provides nourishment to the developing embryo further. Now the zygote develops into embryo and embryo into a sporophytic plant body. Plant life cycles and alternation of generations. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gamete-producing haploid gametophyte and spore-producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. Sporophytic haplontic life cycle. Sporophytic generation is represented only by one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plants is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae, such as Volvox pyrogyra and some species of Chlamydomonas, represent this pattern. Diplontic life cycle. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. Gametophytic phase is represented by single to few-celled haploid gametophyte. This kind of life cycle is termed as diplontic. An alga fucus represents this pattern. In addition, all seed-bearing plants, that is gymnosperms and angiosperms, follow this pattern with some variations wherein the gametophytic phase is few to multicelled. Haplodiplontic life cycle. Bryophytes and pteridophytes interestingly exhibit an intermediate condition, haplodiplontic. Both phases are multicellular. However, they differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte, and it alternates with a short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular saprophytic or autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them such as Ectocarpus, Polysiphonia, kelps are haplodiplontic. Fucus in alga is diplontic. Life cycle patterns. Haplontic. Dominant phase is gametophyte, that is N ploidy. On gametogenesis, produce gametes, and after syngamy 2N zygote is formed, which is dependent and short-lived and performs meiosis to give spores, again a haploid condition. Spores after mitosis give rise to gametophyte plant body. Example are most algae, Volvox pyrogyra chlamydomonas. Diplontic life cycle. Here gametophytic phase is few to multicelled, short-lived and dependent. Sporophyte, that is 2N, is the dominant phase, which on meiosis gives rise to gametophyte N. Gametophyte on gametogenesis produce gametes, which on syngamy forms zygote 2N phase again. Zygote germinate to form a sporophytic plant body. Haplodip examples are fucus, angiosperms and gymnosperms. Third life cycle is haplodiplontic. This is an intermediate condition where both phases are multicellular. However, in bryophytes, the multicellular sporophyte is totally or partially dependent on the gametophyte for its anchorage and nutrition. So in bryophytes, gametophyte is the dominant phase, whereas in pteridophytes, the diploid sporophyte is the dominant vascular plant body, which alternates with multicellular saprophytic, independent but short-lived haploid gametophyte. Here gametophyte is independent but is short-lived, so sporophyte is the dominant phase. Sporophyte 2N on meiosis gives rise to spores. Spores on germination form gametophyte. Gametophyte produce gametes by gametogenesis. Syngamy occurs, zygote forms. Zygote germinate to form sporophyte. In bryophytes, N phase is dominant and 2N depend on N partially or totally for food. In pteridophyte, both phases are independent but sporophyte is dominant. Summary. Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Algae are chlorophyll-bearing simple thalloid autotrophic and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely Chlorophyceae, Phaeophyceae and Rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes, which may show isogamy, anisogamy or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like, prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like and stem-like structures. Bryophytes are divided into liverworts and mosses. Plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axis bearing spirally arranged leaves. The main plant body of bryophyte is gamete producing and is called gametophyte. Bears male sex organs called antheridia and female sex organs called archegonia. Male and female gametes produced fuse to form zygote, which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant body is the sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissues. Sporophytes bear sporangia, which produce spores. The spores germinate to form gametophytes, which require cool, damp places to grow. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of male gametes to archegonium. A zygote is formed after fertilization. Zygote produces a sporophyte. Gymnosperms are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed and therefore these plants are called naked-seeded plants. The gymnosperms produce microspores and megaspores, which are produced in microsporangia and megasporangia borne on the sporophylls. Sporophylls, microsporophylls, and megasporophylls are arranged spirally on axis to form male and female cones respectively. The pollen grain germinates and pollen tube releases male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, zygote develops into embryo and the ovules into seeds. In angiosperms, the male sex organs, stamen, and female sex organs, pistil, are born in flower. Each stamen consists of a filament and anther. The anther produces pollen grains. that is male gametophyte after meiosis. Pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or embryo sac, which contains the egg cell. The pollen tube enters the embryo sac where two male gametes are discharged. One male gamete fuses with egg cell syngamy and the other fuses with diploid secondary nucleus, triple fusion. This phenomenon of two fusions is called double fertilization and is unique to angiosperms. The angiosperms are divided into two classes, dicots and monocots. During the life cycle of any sexually reproducing plant, there is alternation of generations between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycles, haplontic, diplontic, and intermediate. Pollen grain is the male gametophyte and embryo sac is the female gametophyte. Levels of the following: protonemal cell of a moss is N, primary endosperm 3N, nucleus in dicot, leaf cell of a moss is N, prothallus cell of a fern N, gamma cell in marchantia N, meristem cell of monocot 2N, ovum of a liverwort N, zygote of a fern 2N. Group of plants that bear archegonia are bryophyta, pteridophyta, and gymnosperms.